Okay, good day. So this is Mrs. Tole from Emerging Private Tutors. Today we are looking at DNA code of life. So this is what you find in paper 2, life science paper 2. So we need to understand that DNA is very important in our life because it provides the code for the synthesis of proteins, okay, which is vital in our lives. Now, we're going to look at the animal cell here, just for you to understand where we find the DNA and also understanding the animal cell itself. Now, so we have a nucleus, which is double membraned. So let's start with the cell wall, cell membrane, sorry. So we've got a cell membrane, and inside the cell membrane, there is a fluid inside the membrane, inside the cell membrane, which is cytoplasm. So cytoplasm is outside the nucleus. So the nucleus is also part of the organelles that you find in the cytoplasm. So in the nucleus, when you look at the nucleus, you see that nucleus is double membraned. So the outer membrane of what we call nucleus is double. So we call them double nuclear membrane. And inside the nucleus, we have what we call the nucleolus, okay? Then also, we have also our DNA, which is your chromatin network. So we have got a chromatin network here, which consists of your DNA. Then we have the ribosomes. So this is what we call endoplasmic reticulum, and we have with the ribosomes here, which is your site of protein synthesis. So this is your site of protein synthesis. We're going to look into that, protein synthesis. So then we also have the mitochondria. Then we also have these pores, which are what we call nuclear pores, the opening on the nucleus, the opening on which will actually be used as a what we call as a passage for a messenger RNA. Messenger RNA. We will look into that as well. Now, so we have two types of nucleic acids which assist in the protein synthesis, which is your DNA and your RNA. So DNA is deoxyribonucleic acid, then your RNA is ribonucleic acid. Then where do we find our DNA? We're going to start with looking into what we call into DNA, and your DNA we find it in the nucleus, is in the chromatin network, and what is it? So we have the chromosomes here. Chromosomes in the what in the nucleus. Then in the mitochondria, it's for both animal and plants. So animal and plants. So you find this in the mitochondria of animal and plants. So we call that mitochondrial DNA. And also chloroplast, which you only have in plants. Then chromosomes are long, thin thread-like structures composed of DNA that is wrapped around what we call proteins called histones. Okay, so let's look into our next slide. We have what we call a gene. A gene is very much, you need to understand the definition of a gene, and you need to understand it very well. Gene is a segment of DNA. So you're saying a small segment of DNA that controls an inherited characteristics or determine the appearing and functioning of an organism. So each gene carries a code for the synthesis of a particular protein. So we are saying genes, they carry information for synthesis of a specific protein, because your DNA contains all the information for synthesis of all the proteins in our body. But now we are saying for each protein, we have a particular gene that code for that. Then who formulated the double helix of what we call of DNA is James Watson and Francis Crick. I think this also will pop up on the what we call on the question 1 on the multiple choice. So you just need to understand that. Now, structure of DNA. DNA consists of 2 strands. So we say DNA is double helix. It's double helix, and when I'm talking of the helix, it's coiling. It's coiled. So and the DNA is a polymer. So meaning to say it's a very long chain made up of monomers, which are your single blocks, which we call nucleotide. So when you're looking at a nucleotide consists of 3 things. It consists of what we call a phosphate group. Phosphate group. Then it consists of what we call deoxyribose sugar. Deoxyribose sugar. Then also, what is it? A nitrogenous base. Nitrogenous base. So this nitrogenous base, we've got two types, we have four different types of nitrogen bases on the nitrogen base. We have got 1, 2, 3, 4. So we've got what we call thymine. We have what we call adenine. We have what we call cytosine. And we also have what we call guanine. So these are the four different types of what we call nitrogen bases that we have on the DNA nucleotide. And each nucleotide, we are saying we have a phosphate group, then we have it's connected to a deoxyribose sugar. So 1, 2, 3, 4, 5. Then here we have our base. So each nucleotide consists of a base. Here is our base, here is our sugar, here is our phosphate group. That's what we have there. So we've got a phosphate group, we have got what we call a sugar and a base. So that's what we call a nucleotide. This is one nucleotide. So this is a building block of what we call of your DNA. Now, when you look at those what we call the nitrogen bases, we have got what we call purine bases. Those are large molecules, adenine and guanine. And we have cytosine and thymine, which are smaller molecules, which you call pyrimidine. So you need to remember this as well in case of what you call your multiple choice. Now, there are four different types of nucleotides. So since we have got different, four different nitrogen bases, that will also give us four different nucleotides. So we are saying we have got the one, we have first one with what? With 3, 4, 1, 2, 3, 4, 5. And here we are saying this have got what we call thymine. So thymine is capital T, adenine is capital A, cytosine capital C, guanine capital G. So here we are saying we have got the one for adenine. We have got another one, 2, 3, 4, 5. And we are saying we have the other one for guanine. Then we have got the other one, which is the cytosine, then the last one, which is your thymine. So that's what we have. So we are saying we have different types of four different types of nucleotide. That's what we have. What is it on DNA? Okay. Now I want to understand the formation of DNA. How is it formed? How is your DNA formed? So we are saying deoxyribose of one nucleotide forms a bond with the phosphate of another. Then two strands are formed with alternating deoxyribose molecules and phosphate groups. Each rung of DNA ladder is formed by linking of two nitrogen bases and a large purine base and a small purine pyrimidine base. So here should be pyrimidine base. Then with each base pair is joined by a weak hydrogen bond. Specific base pairing, we have got adenine connecting only to thymine and guanine only connecting to cytosine. Then two hydrogen bonds for A to T. So we have got two hydrogen bonds connected there. Then we've got three hydrogen bonds for G and C. So we have three hydrogen bonds there connecting. The number of adenine are equal to number of thymine. It has to be balanced because remember they do pair, and base pairs occurs in any sequence. Then sequence of bases provides a code that gives instruction for synthesis of proteins. And we are saying this sequence of bases is what we call genetic code or the code of life. And DNA strands, we are saying they are complementary. So what we are saying here, let's just give an example here of what I'm talking about here. So when I'm talking of a double-stranded, this is what we mean. Okay. Then we have our another phosphate again that is connected. Okay, then we have our base here. So we can say this base can look like this. Then maybe this one can be like this. Okay, then here what we have here, we have something like this. We have another base of another what we call nucleotide. Okay, with its phosphate. And when you connect another nucleotide with is also what we have here. We have got another base here, which will connect to that. So here, what we are simply saying here is adenine only pairs with thymine. Then guanine only pairs with cytosine. That's what we're trying to say here. And we are saying, how many bonds do we find here? We find 3 of them. Then here we find only 2. That's what we have. And this is what we call hydrogen bond. So your hydrogen bond, H bond, then you have got a phosphate group here, and we are saying it's double, meaning to say, you can say it looks like a ladder. We have got one strand here. This is one strand, this is another strand. So we've got 2 strands that are actually connected together, bonded together with hydrogen bond. So this is a strand that goes down, a strand that goes down separately, and we are saying we can separate these 2 strands by breaking of this what called hydrogen bond. So the DNA strands can be separated through what called when the hydrogen bond breaks. Then we are saying, what is the role of DNA? The role of DNA, it carries genetic genes. It carries genes, which is your genetic code, which are what called with a specific sequence of what nitrogen base. So we are saying these genes carries information or what called code for the synthesis of a particular protein, or for synthesis of what called controlling a particular characteristic in an organism. Now, DNA replicate, make identical copies to ensure that genetic code is accurately transferred from one generation to another generation. We'll look into DNA replication just now. Then we have also what we call, this is what we call coding DNA, the one which code for protein synthesis. But we have also non-coding DNA, which doesn't play what we call any part in proteins, which doesn't code for protein synthesis. But what does it do? It regulates and controls expressions of genes in the coding DNA. It also protects the genes from mutations and controls the process of copying genes during transcription. Now, let's look into DNA replication. Now, DNA replication, when a DNA make a copy of itself, that's what we call replicating. So DNA replication is the duplication of your DNA. So you're making double DNA now. So you're saying when you have got one DNA, you're making another what you call a DNA, which is identical to the previous one. So here we're saying DNA replication is a duplication of DNA molecule to form 2 identical copies, which are replicas, and it takes place during interphase of cell cycle. Now, 2 identical units called chromatids are formed and are held together by what called a centromere. So we are saying here, we have our DNA here, we have a chromosome, so that what call, we have a chromosome. Remember your DNA, it's like this. So we have our, these are the histone proteins. So this is your DNA. This is the histone protein. And what do we call this whole thing here? This is what we call a chromosome. So it's your DNA wrapped around a histone protein. So here we're saying we have got a single strand here. Okay, let's say a single strand here. Then this single strand, after replication, we have what called the centromere here that touches, that connect, link up this. So this one here is what we call a chromatid. A single one is a chromatid. And now we are saying we are forming 2 identical chromatids. These are 2 identical chromatids that we have here. These are 2 identical chromatids. So when DNA replicates, it replicates and forms and make a copy of itself. Here what we have is a 1 chromatid here and forms a connect what we call after replication, it have another chromatid which is identical to the first one. And there are 2, these 2 are connected by what we call a centromere. So I think you understand this when I'm also doing cell division. Now, process of replication. So what happens now for what call for replication to take place? What actually the steps that occur during replication? You need to understand this because it can also come to describe what called when you ask you to describe DNA replication. So what happens now? Remember what we said? We said our DNA is coiled, it's double helix, it's coiled. So when it's coiled, it needs to unwind first. So when it unwinds, it becomes what you call a straight. When it unwinds, it becomes straight. So it has to unwind. Then when it unwinds, it becomes straight and forms. It becomes shows you something like a ladder like this. This is what you actually have. Okay. So we are saying weak hydrogen bond break and two DNA strands unzip. So here we are saying when after unwinding, what happens is what called the unzipping. Unzip. So when it unzip here, what happens now? We are saying it separates like this. So it's actually exposing the bases now. It's exposing the bases. I'm just going to give you an example here of maybe let's say A, T, C. Then this one is going to be the complementary. It's going to be T, A, and G. That's what we have here. So when it's open, that will be unzipping. Now, when it's unzipping like this, each original DNA strand acts as a template. So here we are saying one of them will act as a template. Each one of them will act as a template. Then we are saying on which it complements forms. Then free floating nucleotides in the nucleoplasm build a complementary DNA strand onto each original two strands. So we are saying the free floating DNA strands here will come and say what called the DNA nucleotides will come here. For example, here we have our T coming here, the one T coming here, connecting here, and we have what called A coming here, connecting here, and these two will connect it together. Then it forms another strand, new strand. So enzyme DNA polymerase controls joining nucleotides. Then two identical DNA copies are formed, each comprising of one old strand and the other one the new strand. So we are saying in the end here, what we have as a result here, we have something like this, a new strand here, a new DNA also that side. So we have A connected to another one here. This is a new strand now, which is your T, which is your T, which is your A, which is your C, which is your G here. So that's what we have here. And on the previous one here, on the other one here is T, and this A, and this G. So we have this new strand now that comes in, will connect here with A. Then we'll have T here, we'll bring also C here. So you can see that these two are actually identical. That's what we're having here. So one is an old, so this one is old strand, this one is these two are a new strands. That's what we're saying here. So in the end, we have got two identical DNA strands. Okay, let's move on to the next chapter. Now, what is important of DNA replication? Now we are saying your DNA doubles. DNA is doubles, replication is doubles, it doubles the DNA. Now, when it doubles the DNA, it ensures that the daughter cells, when they split, when they, or during what we call mitosis, or during what we call meiosis, when they split, will have what we call enough DNA for each of the what we call, or of each of the daughter cells. So it ensures that we have enough DNA on the what we call next generation, from generation to generation. So it ensures that enough DNA is available to be passed on from generation to generation. So it ensures that during mitotic division, cell division, daughter cells have same genetic information as the mother cells. So when you have the mother cell here, it is split into two. So when it's split into two, make sure that when you've got DNA, so let's say this is our available DNA here. After splitting it, what we need to have each cell here. These are what we call daughter cells. This is the mother. These are the daughter cells. So these daughter cells, what we have here, we have got, they must have the same DNA as the mother, and also they must be the same, they have enough, the same DNA. So for this to happen, it means that the mother has to double its DNA. When it doubles its DNA, it makes sure that it will be available to be split on the two daughter cells. So you find that this DNA is similar, is identical to each other and also to the mother cell. Okay, so I think this is actually clear to you. But now we see that this DNA sequence, the sequence of your of your bases, is actually unique from one individual to the next individual. So we are saying this what we provide what we call DNA profiling. So that's why now you see that when maybe you are a suspect, they collect your they actually take your your fingerprints. So or when you're taking ID, you need to be taking what called the fingerprints, because it's unique on each of each individual, except identical twins. So identical twins, they do have what called same DNA sequence. Now, because identical twins is just one what called fertilized egg, which have got same DNA, which split. So after splitting, you find that it's identical. So you find the DNA that is identical on both on the offspring, or should I say on the children. Now, here what we have here, DNA-based sequence differ among individuals except identical twins. I think I've explained that. Then also DNA-based sequence gives a unique barcode pattern known as what called DNA profile or DNA fingerprint. DNA is extracted from skin, blood, saliva, semen. That's why you see that when there's a crime scene, on a crime scene, you find that those for forensics, those working on the forensic will be collecting hair, will be picking up cigarettes, will be, what call brushing the saliva, on also collecting what on the steering. They're trying to take the sweat and everything. So that's what we have there. We're collecting different what call types of samples. These are the ones that will be used to extract what the DNA of that individual that have been smoking the cigarette, or that was at the scene and left maybe some hair there. Now, uses of DNA profiling. Where do we use the DNA profiling? DNA is used to identify crime suspects and also a proof of paternity. I think you find that if you are not sure that your child, you are a man, maybe a child, you don't sure that it's your child, you can go for paternity test, and it's actually the DNA that is being tested compared for what call if your DNA, if you have contributed on the child or not. Now, to determine the probability and cause of genetic defects, and also tracing missing persons, establish compatibility of tissue types, for organ transplant. Then what about disadvantages? Because there are also challenges here when you're doing DNA profiling. Inaccuracy of DNA profiling process leads to errors. Then also human errors in interpretation of the results. Data may be used to discriminate against an ill person. Cost of profiling may exclude poor communities. Forensic labs do not confirm or to uniform standards. So we are saying there are more challenges here, because sometimes you find that someone can actually pick up your cigarette that you have smoked and plant it and throw it maybe on a crime scene. When they take that information, they are going to think that you are actually one of the suspects, but you are not. And we are saying also this can also be biased, human errors as well, because remember, you'll be dealing with these people that process this thing are humans as well. So they can make mistakes and actually give false results. Now, DNA samples may be planted at the crime scene, and small samples of DNA may be also identical in suspects. Now, so we're done with DNA for now. Now, we look into RNA, which is your ribonucleic acid. So we're looking at the location of your RNA. It is nucleus. And also, what about the cytoplasm? It's also found in the cytoplasm and also ribosomes. What about the structure? The structure here consists of a single strand. So RNA is single-stranded, DNA is double-stranded, okay? But still here, we still have the same, same nucleotides, except one here. We are saying on the nitrogenous basis, the nucleotide is made up of what? We have got the phosphate, and we have got the sugar here. Then what do we have? We have got the base. So here what we have on the sugar here is no longer a deoxyribose sugar. Remember, we said we have a deoxyribose sugar on the deoxyribonucleic acid, but on the ribonucleic acid is what we call a ribose sugar. Then here is still the phosphate group. But on the basis here, what we have something new here. We have now what we call uracil. Uracil is replace thymine in the DNA. So in RNA, whenever you see RNA, you must think of uracil replacing thymine. Then we no longer have thymine in that side. Now we have got other ones are just the same: adenine, guanine, and also cytosine. Okay, then formation of RNA nucleotides join when ribose of one nucleotide attaches to the phosphate group of another to form a single long strand. 4 nitrogen bases, adenine, cytosine, guanine, and okay in n ratios. Here now, because there is no pairing of bases, there is no pairing of bases, so they okay in any sequence, and they actually not equal. Like for example, in the DNA, we know that adenine, number of your adenine are equal to number of your thymine, because remember they are pairing. But here there is no pairing, so they can okay in any sequence, in any form. Okay, so okay, let's move on. Types of RNA. We have got different types of messenger RNA. We've got the first one is the messenger RNA, and the second one is transfer RNA, and the last one is ribosomal RNA. So this is the one that is used to copy the DNA. So used during transcription. So it carries the message. It carries the code. Carries code. Genetic code to the ribosomes. Because we are saying, I will explain that later. Transfer RNA, this also have got carries amino acid. It does carries amino acid to the ribosomes. So what's good about transfer RNA? You find that it carries one side, it have got bases, nitrogen triplet bases, and the other side it have got amino acid. So we have got a transfer RNA. I'm just going to draw a sketch here. So we have got the bases here, and we have got amino acid here. So nitrogen bases that you find here, let's just say A, maybe A, C, G. Then here we've got amino acid. Then you've got ribosomal, RNA, which is also part of the ribosomes. You find it in the ribosomes. Now, different types of between DNA and RNA. We can talk of the sugar. We're saying deoxyribose sugar. Here we've got ribose sugar. Then we have got, what else can we talk about? Double-stranded. This one is single-stranded. Here we've got base of thymine. Here we've got uracil. So these are major difference that you find in terms of your DNA and the RNA. Okay. Now let's move on to what's called protein synthesis. Protein synthesis. This is a very important part of this chapter. Protein synthesis. We have protein synthesis now is split into. Let me just, okay, I think I skipped here. Protein synthesis is a process whereby proteins are manufactured in living cells, and it's controlled by DNA and the messenger RNA and the solvent in the RNA. So protein is a polymer with amino acid as a monomer. So here the building blocks are amino acids. Then specific type of amino acids are joined in a specific sequence to form a specific protein. So we are saying we have your DNA, part of the DNA, which have got a certain sequence, and that second sequence code for the synthesis of a specific protein. And that segment of that DNA is what called the gene. Remember that. DNA provides the genetic code that determines the type and the sequence of amino acid. Only a small segment, which you call a gene of DNA strand, that is involved in the synthesis of one protein. One base triplet, 3 consecutive bases. So we're saying base triplet, I'll explain this later. 3 triplet bases. So a base triplet is your 3 consecutive bases, which provide code for a particular amino acid. So we're saying one base triplet is equal to your 1 amino acid. Okay, that ratio you must understand it. So we're saying 1 amino acid, 1 amino acid as to 3 bases. So this is the ratio that you must know. 1 amino acid as to 3 bases. Now, base triplet sequence determines amino acid sequence. So how they are sequenced is also determine also how your amino acid are sequenced. So protein synthesis occurs in 2 main steps, which is the transcription. When we talk about transcription is a rewriting of your DNA. Then translation is triplet base sequence to amino acid sequence. That's what we're saying here. Then now, let's look into transcription. So we're saying transcription, your DNA is too big. Your DNA is too big to fit on the nuclear pore here. That's what we do. That's what we see. The DNA can't fit here. So what do we need to do? We need to copy the information on the DNA and take it to the ribosomes where protein synthesis is okay. Now we're saying, now what is called there? This is what transcription. Transcription process where messenger RNA obtains the code for protein synthesis from DNA. Now, how does the process go? So we're saying a segment of DNA serves as a template for building messenger RNA. So we're saying a segment of DNA serves as a template, and DNA has to unwind, weak hydrogen bond break, and DNA unzip, and where the gene is located. So it has to unzip what only the gene is. Because remember, we're saying your DNA is very long, it's very long. So it's saying only the part, one part is opened. So this is the part that is opened and exposed. The bases are exposed here. When these bases are exposed, this is what called the gene that we're talking about. So, but this whole thing is the DNA. Here is closed, it's still closed here, and the other part here is still closed as well. But we're saying this small part here is actually opened, and what happens now is that your small messenger RNA will come and copy that information. Once you copy the information, it leaves the nucleus. I will explain that as well. Now, enzyme RNA polymerase controls the process. Free-floating RNA nucleotides in the nucleoplasm are used to build the complementary messenger RNA strand. Then, in RNA, the thymine is replaced with uracil, specific base pairing A to U and C to G. So there's always specific base pairing, guys. You must understand that. Adenine always pair with uracil, and cytosine always pair with guanine. A single messenger RNA strand is formed. Messenger RNA leaves the nucleus via nuclear pores to ribosomes in the cytoplasm. Messenger RNA base triplet, which you call it the codon, codes for a particular amino acid. Okay, then we go translation now. The translation now, we are moving from, now we are on the ribosomes where the protein synthesis occurs. Now, here now we have got messenger RNA base sequence is translated to amino acid sequence. Messenger RNA from nucleus attached to the ribosomes. Then messenger RNA provide the code for linking of amino acids to form specific protein. Your transfer RNA picks up amino acids in the cytoplasm and it brings them to the ribosomes. Then transfer RNA, you've exposed the base triplet, which you call anticodon. So you've got messenger RNA, you've got a codon, transfer RNA is anticodon. Then the codon of messenger RNA and anticodon are complementary. So they have to pair, they are complementary. Then transfer RNA places amino acid in a sequence, and the amino acid forms a peptide bond with adjacent amino acids to form a polypeptide chain. Now transfer RNA breaks down into breaks bond with amino acid and picks up another amino acid of the same type. A polypeptide chain of amino acid forms a specific protein. So I'm just going to do a sketch here of so that you can understand what we're talking about here. Now we're saying here, let's have a nucleus here. Let's have a nucleus here, and in this nucleus here, okay, in this nucleus here, we are having a DNA. So let's have a DNA here. Let's have a DNA. We have our DNA, and let's open it. This is our gene. Let's open our gene there. Then let's have our DNA continuing vertical like that. That's our DNA there, and it continues up to what coils like that. Now, here we're saying this part here is what we are exposing now. Okay, this is what we are exposing now. And we are saying we are not worried, let's not worry about the other side, because remember it's double stranded. Now we are saying, let's say we have got A here, let's have T, let's have C, let's have A, or maybe let's have G here, let's have C, let's have T, let's have A, let's have G, let's have G, like that. So what happens now is that in here we have, in this article, this is a nucleoplasm. Nucleoplasm. And in this nucleoplasm, we have got a free nucleotide floating here in the nucleoplasm, and that will come here and form and pass with what? It comes and pairs with your messenger, your DNA strand here. And this is our messenger RNA. This is our messenger RNA that is here to collect the information. It's here to collect the information. And what happens here when it's pairing here is going to pair with U. Here is going to pair with A. Here is going to pair with G. Then it comes here, it's pair with C. Then here is G. Then here is A. Then here for this one here is U. Then here is C. This one is C. So that's what we have here. So that's our messenger RNA now. Now, once the messenger RNA copies this code, this is the code that we're talking about. It carries it, comes out to what you call the ribosomes. Now, here now, your ribosomes, this is your site now for protein synthesis. Ribosomes. Now, on the ribosomes, it comes here and put itself, it's a line self in there. So we take again the same, let's take the same bases here. So what are those bases? We have got U, A, G, okay. Then we have got C, G, A, then we've got U, C, C. That's what we have there. So this is your messenger RNA. Okay, this is your messenger RNA now aligning itself in the what you call in the ribosomes. It's in the ribosomes now. Now, what happens now is that these triplet bases, these three bases that we're having here, these three bases here that we're having here, we call it the codon bases. So it's messenger RNA triplet base. Triplet bases. So there are three of them. So we call them codon bases. Codon bases. So there's the codon. Now we have got another guy here because remember this is a cytoplasm. In this cytoplasm, transfer RNA is busy here. We have transfer RNA and transfer RNA. Remember what we say, transfer RNA, one side is have got what? It's have got, let's just align one transfer RNA here. And this transfer RNA carries amino acid. Let's call that amino acid, let's call it valine. Then here we have got a complementary, it has to complement because transfer RNA will come here where it's complementing, where it have got, it can complement with any of the triplet bases. That's where it sticks. It doesn't go anywhere. It has to come here. So for this one here, it has to be what? U, it has to be A. This one has to be U. This one has to be C. So come here with valine. And when you have that, so let's just show you something here. And this is the triplet bases. We call them anticodon. This is the transfer RNA triplet base. So transfer RNA triplet base is what we call anticodon. That's what we have here. Then here we have another transfer RNA that comes here and attaches here. But this is a temporary bond. This is a temporary bond here. The temporary bond here. This is a temporary bond. Temporary. Now, this temporary bond is just for us to link the amino acid. That's what we're saying here. Just for us to link the amino acid. So let's have another transfer RNA is carrying amino acid. Let's call that amino acid, maybe let's call it what? Let's call it methionine. Okay. Then here we have got G, we have got C, we have got U. Remember? Complementary base pairing. Then here we have another transfer RNA. Okay, then another transfer, what is it? What do you call it? Another, so let's just call it proline. So this is another amino acid. So these are what we call amino acid now. So what we are having now is that we are having amino acid sequence. Let me just show you here. It's U, and U is pairing with what? U is pairing with A, and C is pairing with the G, is pairing with the G here. That's what we're having here. So what we're having here is that this is long, and also this will also be long. So what happened now is that this forms a temporary bond here. When it forms a temporary bond, it's moving there. Temporary bond, and when it forms a temporary bond, it wants the amino acid to link up. So here we have got amino acid sequence now. And this amino acid sequence is following the sequence on the base, the sequence base, which is the code that we obtained from what? From the DNA. That's what you're having here. So we're saying now we have got amino acid, what we call this sequence here that we're having of amino acid. And what we have here between amino acid is what we call a peptide bond. So amino acid will be forming peptide bond underneath here. Once they form the peptide bond here, this one what called release the amino acid and detach itself. Once it detaches itself, where does it go now? It comes and picks again another what you call another amino acid in the what in the cytoplasm. Then it brings it here and look for where it can match. Once it matches, it forms the amino the sequence here until we form a polypeptide chain. Polypeptide chain, and that polypeptide chain has to have more than 50 what we call amino acid, and that will give us a what we call a specific what that will result in a specific protein. Because remember, when you combine your amino acid, proteins are building what we call it's actually amino acids are building blocks of proteins. So you must know that when you combine these amino acids, what more than 50 of them, we are now having a protein, specific protein that performs specific functions in the body. So that's what you need to understand here. So what the major thing is that to know how to able to recognize that this is my ribosome, this is a messenger RNA, this is what we call the codon, this is the anticodon, and where is it? This is a peptide bond, this is an amino acid, this is when it's these are amino acids. Amino acids. That's what we're having here. So we have amino acid sequence down here, and we've got the base sequence up here. That's what we have. Okay. So let's just try to check if we can have maybe one question there to look into. All right. Now let's have question 1.4. This is October. Is it no, June 2018? So as we can see here, what we have here is that the diagram below represents the process of translation. Okay, I just forgot something here. This part here is transcription, where we are copying messenger RNA, the code to the messenger RNA. Then in this part here is what called the translation, where the base sequence is translated to amino acid sequence. Once we have our protein, then the process is done. Okay, so let's check here what we have here, process of translation. Now, name organelle X. Organelle X is our ribosome. Guys, this is our ribosome. Organelle, that's where the site of protein is. Okay, then molecule W and Y. So W is your messenger RNA. What about Y? Y. Once you see Y, once you see something which contains two parts here, so we have amino acid and the bases here. So that's our transfer RNA. Okay. Then monomer of molecule W. Monomer of molecule W. Monomer here is this molecule W is an RNA, messenger RNA. So what is our monomer? It's nucleotide. Nucleotide. Okay, now where in the cell is organelle X found? Organelle X, where is organelle X? That's our ribosomes. Where is it found? Cytoplasm. Okay, where is molecule W found? Where is the molecule W? It's your what is it? It's your, where is it found? Where in the cell? Where do we find it? We find this molecule W is your messenger RNA. We're finding it in the ribosomes. Now, but the messenger RNA here, okay, then let's move on to the next one. We are saying insulin is one of the hormones responsible for control of blood glucose levels in humans. It is made up of long amino acid chains, A and B, which are joined, A and B, which are joined here. So they are joined together to form your insulin. Chain A is made up of 21 amino acid, and chain B is made up of 30 amino acid. Then what do we have here? The diagram below represents amino acid. Now, name the process whereby insulin is produced in a normal human cell. It's what we call protein synthesis. Okay, then identify bond X. These are amino acid, guys. These are amino acid. What is that? Is peptide bond. Okay, then how many nitrogenous bases in a DNA molecule code for amino acid in chain A? Now we're saying how many nitrogenous bases code for amino acid in chain A. Now we are now saying, remember, amino acid, S2, base. We say it's 1s to what? 3. 1s to 3. Now, how many amino acids do we have in chain A? 21. So how many should we have bases now? 21 times 3. What is 21 times 3? 63 bases. So 63 bases. That's the answer. Then the table below shows the messenger RNA codon that code for some amino acids. Now, name the nitrogen base represented by G in the form in the messenger RNA codon CAG. Now we have got our codon and the amino acid. Okay. Now we are told that name the nitrogenous base represented by G in the messenger RNA codon CAG. Where is CAG? CAG here. Now we are told nitrogen base. What do you call that? Guanine. So G is guanine. Now represent the chains. Read the chains from left to right and give the chain from left to right. Now give the codon for the fourth amino acid in chain B. Fourth amino acid in chain B from left, 1, 2, 3, 4. Now codon for that chain B, it's what you call what is it here? Glutamine. It's glutamine. And what is the codon for glutamine? Codon for, just speaking, guys, here is CAG. You're not doing any job here, you're just identifying them. Okay, so that's glutamine. Then read, saying, okay, give the DNA-based triplet that code for the first amino acid in chain A. So in chain A, what do we have? We have got what you call GL, okay, glycine. Now in the glycine, we are looking for, this is the codon, the messenger RNA. So glycine is G codon, is GA. What about the DNA one? The DNA should be what? It should be CC for E. For A, remember, we no longer have, it's DNA now, we no longer have U, we have got T. So this is CCT. So it's CCT. Now give the anticodon for valine. What does it, which pairs with valine? What is valine here? We have got valine is GUA. Now GUA, what does it pair with? GUA. So codon, anticodon. So we have got codon is GUA. Now what about the anticodon? It has to be C. When it's U pairs with what? U pairs with A and A pairs with the U, because remember, this is still transfer RNA. Anticodon is for transfer RNA. Remember, transfer RNA. That's the anticodon. That's where you find the anticodon. Okay. I hope this was helpful. Meet you in the second lecture. All the best in your studies. Thank you so much.